Okay, let's continue on. One thing I did forget to say about glycolysis in the last video is that glycolysis does not need any oxygen to happen. So this has nothing to do with oxygen. You can do glycolysis with zero help from oxygen. And the other thing is when you break the sugar into two and harvest the electrons, you also, a little bit of energy escapes, and that can be used to make a little bit of ATP. So it's made by a sort of separate side process. So a little bit of ATP is made in glycolysis, but not very much. Okay, and glycolysis. Now we'll talk about the next step, which is the transition. All right, so we've got these two molecules of pyruvic acid or pyruvate that each have 1, 2, 3 carbons in them. What we're going to do now is move these molecules from the cytoplasm into the inside of the mitochondria. That's why we call it the transition, because you're literally transitioning locations from outside the mitochondria into the mitochondria. And you have to cross through two membranes. You've got to cross the outer mitochondrial membrane and the inner mitochondrial membrane all the way into the matrix. So when that happens, we're going to do one at a time. We're going to take our pyruvic acid. It's going to go through the cytoplasm. It's going to go through the first membrane, and it's going to go through the second membrane. It goes through a little channel, and when it goes through, one of these carbons is broken off like this. All right, so what we've got here is a single carbon that we broke off of our pyruvic acid, one carbon. So we can harvest the electron that was holding it in place. Guess where we're going to put that? In another taxi. There it goes. So now it's NADH. We're going to put that there. And this carbon that was ripped off of glucose is now just a single carbon. It's basically carbon dioxide, and that's what we breathe out. So off it goes. We don't need that anymore. And what we have left is two carbons stuck together. This is actually called acetyl-CoA. So I'm going to write that on here. The pyruvic acid goes through what's called the transition into the mitochondria. Can't reach over there. And it turns into what we call acetyl or acetyl-CoA. And a single carbon dioxide is also pulled out. All right. Acetyl-CoA is a 2-carbon molecule. Look, there you go. There's just 2 left, right? And the third one just got stripped off and breathed out. So we're just going to breathe that away, up your nose, off it goes. All right. So this is what happens at the end of the transition. You've got acetyl-CoA inside the mitochondria, ready for the next step. Now, of course, we had to do that twice because we got the other pyruvic acid, because we had 2 of them. So we're going to move the other one into the mitochondria. It goes. We're going to strip off the carbon. We're going to breathe that out. We're going to strip off the electrons, put it in another taxi. We'll use the same one. It's in the taxi. All right. So we've got our electrons being harvested here in taxis. And now all we have left of glucose are these 2 little acetyl-CoA molecules. And this is what we're going to use for the next part, which is the citric acid cycle.